We're going to talk about taste, and we're also going to talk about hearing, and we're going to talk about equilibrium for the second day of week 4. So we're going to start off with taste. Pretty simple lecture, pretty short. So here are 3 outcomes for the day. So when it comes to taste, this is activated via taste buds, which you're all well aware of, or at least have heard of. And as you can see, what's really interesting about taste buds is they have little tiny hairs that stick out that are there looking for or waiting for a chemical that's been dissolved in the saliva to activate them. All right. Now, typically taste buds, they're all chemoreceptors, so they're activated by specific chemicals. Taste buds are typically sensitive to one of the 5 tastes, but if you have a high enough concentration of some specific taste, they'll be activated. So here are the 5 flavors that we can taste. And most of these tastes you should know, or probably already know what activates them. So we've got sour, which is activated by acid. So acid is hydrogen ion, so that's found in like citrus. Salty are by metals. So, think it's like sodium, things that you think of with salt. Sweet is activated by glucose or specific carbohydrates. Bitter is a little bit different in that there's not really a specific thing that we can identify. We just know that flavors that are bitter activate these bitter receptors. And then we've got umami, and this is activated by glutamate. So that's that meaty flavor. If you've ever been to some restaurants that use MSG, that's why it tastes so good. It gives you that meaty, savory flavor. It's important to know that, come on, clicker, there we go, that sour and salty are, when they just open ion channels, whereas the other 3 are G protein coupled receptors. And these G protein coupled receptors, the G protein is called gustation. And what's also interesting is that every flavor is also sensed by smell, right? So I shouldn't say we all, but I actually remember this is probably something that scarred me for life. I hate fish, just despise it. I just don't think it tastes good. And I've always been that way. And my parents are like, you've got to try one bite, blah, 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 blah. And there was one night where I wouldn't do it. And eventually I did. And the way that I was able to survive, so to speak, the eating the fish was when my mom told me that all I need to do is plug my nose and then it doesn't taste as fishy. And so that's what I did. Okay. But as I said earlier, before these receptors are stimulated, these chemicals that activate these taste receptors have to be dissolved in fluid or aqueous solution, so mucus or saliva. So here we've got a diagram that's going to make some people's heads explode, and it's not as crazy as it looks. Okay, so I'm going to walk you through this diagram, and then on the next slide, I've got what's going on in the diagram in words. Okay, so here we've got our G protein coupled receptor, and so we'll have something that's sweet, bitter, or umami chemical activate the receptor. Now notice in this case that we're not worried about alpha. We're not worried about GDP. We're not worried about GTP. We're worried about beta and gamma. And so beta and gamma leave. And what happens is they activate a protein called PLC. And PLC is an enzyme. And what it does is it breaks something called PIP2 into 2 parts. It breaks it into DAG, which are kind of like the tail things here, and then IP3. We don't care about DAG in this particular instance, but we do care about IP3. What IP3 does is it goes and it activates a channel on the sarcoplasmic reticulum, and it allows calcium to be released from inside the endoplasmic reticulum out into the sarcoplasm, which causes depolarization. Okay. Now, just kind of getting ahead of ourselves a little bit, or just something to look forward to, is that calcium and the endoplasmic reticulum, that's going to be really important when we talk about muscle cells. We're not talking about muscle cells right now, but just kind of keep that in the back of your mind. Okay, so let me repeat real quick. Something binds to the G protein receptor, beta and gamma leave, and they activate PLC. PLC takes PIP2, breaks it into DAG, which does stuff that we don't worry about, and IP3. IP3 opens a door on a calcium channel, lets calcium into the cytoplasm, I might have said sarcoplasm before because I always think about muscle cells, but it just releases it into the fluid inside the cell, which depolarizes the cell, and then ultimately neurotransmitters released. Okay, so this G protein is called gustation. Beta and gamma activate PLC. PLC increases the production of IP3 by breaking down PIP2. IP3 opens calcium on the endoplasmic reticulum. Calcium goes inside the cytoplasm, which causes depolarization. So, what do you need to be able to do? These three things. So, what kind of receptors are taste buds? What are the 5 main tastes and what activates each of them? And summarize the gustation pathway.